Okay, my bad. Looks like we chopped that off. Maybe this will be part 2. Okay, so we are looking at the graph now on FRQ Unit 6, AP Classroom. Okay, I'm looking at the graph, luciferase activity here, and I'm noticing that all of the experiments here, because we're looking at what, like 11 different experiments, they have the luciferase gene, but do they have the upstream regulation sequences? Well, they're basically including all sorts of different things, right? So we've got this little +1 thing that is like something upstream. Maybe that's where the promoter binds, something like that. A transcribed region. But then we've got 15 bases, 59 bases, 123 bases, 149 bases, 199 bases, 239. Now, why do I think these are bases? Where did it say that? It doesn't quite say that, but we're talking about DNA. So I'm thinking, what are those numbers? Those just random numbers might be the length of the bases, the number of bases that are included upstream to luciferase gene. Okay, and then it kind of shows how much luciferase you actually get produced there. Okay, in wide variety, actually, pretty interestingly. Okay, now the parent plasmid with non-CD3 gamma active promoter basically says, wow, well, that's the one, that's like max business, right? Because that is like, whoo, it's over 34. Okay. And so if you put in just an active promoter, which I think is just some kind of generalized promoter, you got to remember promoter region allows for transcription to occur. And so that's game time. All right, so now we've looked enough of that, so we got to jump down to the question, okay? Identify both the cellular component and the location of the component that is responsible for producing the luciferase protein from mRNAs transcribed in the plasma containing T lymphocytes, okay? So again, this starts with just general knowledge, okay? We go from knowledge to skills down to theory, okay? So this one's asking us a very simple thing. Stop right there, answer that question, and then we'll look at the explain. But it's saying, what are the cellular components and the location that produce a protein? Yeah, they're asking the specific luciferase protein in specific T lymphocytes, but it's all the same, okay? We've got the nucleus contains the DNA, okay? Although the plasmid is typically not contained within the DNA. So you could say something like, all right, well, plasmids primarily are located in the cytoplasm, and you will have RNA polymerase transcribing the plasmid, the luciferase gene region, and producing the mRNAs. That says the cellular component and the location of the component. Boom! You could keep going and say, all right, and then after the mRNA is produced, then you have the ribosome in order to translate the mRNA into a functional protein, which is very easy tacking on there. And remember, use all of your vocab. Don't just say, oh, well, something reads the DNA and makes a protein. I mean, it's just, that's not the vocab that's going to get you the points, okay? And we got to score up on those chumps that are using no vocab on their answers, okay? Next section, explain, okay? Now, explain is a word you got to pay attention to. It's in bold, so we pay attention to those. Identify is something you could use 1 sentence, 2 sentences, right? Explain is something you minimum 2 sentences, maybe more like 4, okay? Explain what dictates to the lymphocytes the correct order in which the amino acid should be linked to form luciferase protein. Again, this is general knowledge. This is not anything to do with your ability to read the graph at all. So imagine you look at that graph and you're like, geez, Rutherford, I don't understand that thing at all. Go to question A. You could answer and squeeze out some points even if you don't understand what the graph or what the experiment even actually is. You know how to make a protein, okay? So what dictates to the lymphocytes the correct order in which the amino acids should be linked? We're basically talking about 3 letters on the mRNA. That's a codon, and each codon in order is going to say the order of amino acids when the ribosome is translating that mRNA sequence. That's how we know the amino acids, how they should be linked in luciferase protein. You want to say things like primary conformation. You want to say things like polypeptide chain, peptide bonds. More points to you, probably, okay? All right, good. Now we're done with B. We don't even have to look anymore. Okay, identify the independent variable in the experiment described. Now, the independent variable is what you independently decide change, right? So what do you think the scientists in this thing independently decided to change? Remember I said dependent is data. So let's look up the things that are changing. Remember it's a variable, so it changes. What are the things that are changing here, okay? Well, certainly the relative luciferase activity is changing. That's the data, that's the dependent variable. And it depends on what? It depends on the independent variable, which is all these different PCD3 gamma 419, 309, 239, 199, 149, 123, all those different amounts, all those different lengths of the upstream regulatory sequence of the DNA, the different numbers of something, right? You could look in a little more and say, like, what are they talking about? Different amounts of the okay, if you realize that this is DNA and that these are nucleotides, drop that vocab and say that the independent variable is the different amounts of nucleotides in the upstream regulatory sequence of the PCD3 gamma. Okay, now remember, always refer back, use things like PCD3 gamma. Don't just say like the gene. Don't just say like the, or that, or it. Don't ever say stuff like that. Refer to things very specifically, okay? So then it says, that's an identify, so that's one sentence. Then another one sentence, 1 point. Identify the plasmid that was used as a negative control for luciferase activity. So I've had to look this up and figure out what a negative control is. Negative control is something that they use in an experiment when they want to compare to something that should have no response, okay? Right? Like something that is like, okay, so we're gonna check and see if how well all of these things work, but we want to compare to something that shouldn't work at all, right? So we can at least see like, okay, well, these all things have a range here, but then this thing is doing nothing. In a medical study, this would be a placebo, okay? We want to test all of these, these drugs for, lowering your blood pressure, okay? But then we're going to give you another pill that's just filled with air. Well, placebo pills are usually sugar, right? Which should actually do bad things for your blood pressure. But the placebo would be a negative control, okay? So the plasmid that was used as negative control for luciferase activity, that is the parent plasmid. Notice that one up there, that was the lowest relative luciferase activity. It just has the parent plasmid. There's no upstream stuff on that one, and therefore that's a negative control. Then it says justify including the plasmid with the non-CD3 active promoter in the experiments. Okay, so why did they include the plasmid with the one at the bottom that goes all the way to 34 activity that is a non-CD3 active promoter? Why did they use that? That's the positive control. This is a good little lesson on what a negative control and a positive control is. The positive control is something that should have like a huge effect, right? So we have basically the nothing effect, that's the negative control, and then you have the like full-on max effect. That would be the positive control. And then you've got everything in between. And why do you use these things? For comparison. Just to see, are these things closer to the negative control? Are they closer to the positive control? Are they spread out all within, right? That's the point of your positive and negative control. Comparison, okay? I might say like something, if I told you something like, you asked me, hey, how did I do on that test? And I say, oh, you did good. Does that give you any information, right? If I say, oh, the relative luciferase activity for PCD3 gamma 199 is 5. Is that good? Is that bad? Is that high? Is that low? Right? Like 5. What does that mean? You ask how you did on the test and I say good. What does that mean, right? If I say 77, then you know, oh, well, the positive control is 100%, right? That's like the highest I could have got. The negative control is 0. That's the lowest I could have got. So 77, it's like, well, it's much closer to the top. Then you also want to know who sets the curve, or who is the highest score in the class, who's the lowest score in the class. If I tell you that the average score was 65 and you got a 77, then you'd be like, wow, freaking A, that's great, okay? it serves as comparison. That's the point of like negative control stuff. Maybe I say like, okay, so I'm giving the test out to everybody. Okay, you got a 77, and a fourth grader got a 25%. Okay, the fourth grader would be my negative control because I assume that the fourth grader knows like virtually nothing, right? In terms of AP Bio, if I give them a tough test, I mean, give it to a kindergarten, giving it to a chimpanzee, right? That would be your negative control. But then I could also I could give the test to a doctor at the U of A or a professor at the U of A that teaches biology. Now, if they only get an 81 and you get a 77, well, then you know you're really close. The professor is the positive control, and the baby, the infant, the chimpanzee is the negative control. That's the best way I could explain that. Let's jump down to C here. We still haven't even read it. Okay, identify the plasmid that must contain the CD3 gamma core promoter sequence, but the fewest or no negative regulatory sequences. So what are they asking? They're asking for, what's the Goldilocks? What's the best one? Which one gives us the most luciferase activity, but doesn't actually inhibit luciferase activity? So again, don't keep reading that freaking complicated question, multi-part question. Answer that one section. Comes up here, and I'm looking at what's the best one? PCD3 gamma 15. Plain and simple. That's the best one. Okay? That's why. Now, you don't just write that. You have to write it as a sentence. You have to say something. The plasmid that, restate the thing, the plasmid that contains the core promoter sequence but the fewest or no negatory sequences is PCD3 gamma 15. Because in figure 2, you see that the relative luciferase activity is about 9.5. You have to say that. Refer to the figures, refer to the graph, so they know you know what you're talking about and you're not just guessing. You say, I chose this because of this that I see in the graph, and I'm referring to it specifically. Next section. Based on the data in figure 2, which again should be, do you see the language they're using? Use that language. Based on the data in figure 2, describe the most likely cause of the variation in luciferase activity among the cells that contain all these different freaking plasmids, okay? Describe the most likely cause of the variation. Why do they go? Okay? This one's going to be a little tougher, right? So this is the skill part, right? Where knowledge, skill, theory, skill of reading a graph. So, but you do need to have a little bit of basic knowledge for this one that says, well, the reason that it's so variable is that maybe you've got your luciferase gene, you've got your promoter region, but then behind that in the upstream regulatory sequence, you might have some other genes that turn it on or off, or go and sit on luciferase and clamp down and prevent it from being functional. The only way you would know this is based on the lac operon, because in the lac operon there is a sequence way upstream called lac I, and that produces the inhibitor that will go and clamp onto the promoter region and prevent transcription. Okay, so that is a bit of knowledge there. Okay, and that to me is probably the hardest part here. Okay, that's a kind of a shot in the dark for me. So remember, you don't have to get 100%. You just need to do relatively well on this problem. You need to get more points than the national average. That's it. That's how you pass. Okay, so don't ever get bogged down if you're like in a question and it's super hard and it's just like, okay, move to the next one. Calculate, again, this is a skill now. Calculate the approximate percent increase in luciferase activity between cells from 59 to 149. Now I gotta use 149 to 59. Okay, so we need to calculate the percent increase. So I'm gonna write percent, and then I'm gonna scroll up, and I see over here that from 15 to 59, or 59 to 149, thank goodness I wrote it down. 149 is over there right around 4. 59 is right around 2. They chose those because it's easy, okay? So what's the percent increase there? Well, to go from 2 to 4, you have to have a full increase. You have to double that. Now, what is that in percentages? It's 100%, right? If you have 2 and you want to increase by 100%, you would increase by 2. If you have 2 and you would like to increase by 0%, how much are you going to increase by? Well, 0, and you're going to be at 2 still. If you're at 2 and you would like to increase by 50%, what's 50% of 2? Well, it's 1, so then you would go up to 3. If you're at 2 and you want to increase by 100%, then you take 100% of what you have and you increase it, right? Okay, that's, we could talk about that in terms of investment. You want to put $1,000 into the bank and have 100% interest increase over a year. Well, at the end of that, you're not going to lose money. You're not going to stay where you are. How much are you going to get? What's 100%? Well, 100% of what you have, $1,000, is going to be added in, and then you have $2,000. Okay, so it's a double thing. And that's calculated there, round to the nearest whole number. Okay, remember, use all the vocab. Say, for CD3-149, approximately 4 relative luciferase activity. For 59, it's approximately 2 relative luciferase activity. And then show your work and take a picture and send it off, okay? Next, D is where we're going to theory, probably. Predict, that's a theory word. Predict the most likely observed level of luciferase activity if blah, 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 7, 8, 9. Writing it down, 7, 8, 9. Is introduced into non-lymphoid cells, such as a line of kidney tissue. Okay, so instead of putting it in a lymphatic cell, lymphatic system, immune system, we're going to put it in the kidney, which filters blood, which I would have taught you if we were in school. Provide reasoning to justify your prediction. Okay, now, the biggest thing with these kinds of things is if you have a reasonable explanation, justification, if you provide a reasonable reason why you think whatever, then chances are you're going to get some of the points, even if it's not necessarily correct. Now, how are you going to be reasonable? Cite the data. Refer to the graph. Refer to whatever they are putting in here, okay? The observed level of luciferase activity, if we did 789, okay? Well, it says, it's right there. In non-lymphoid cells. Okay, so for lymphoid cells, it was about 3. So you might just want to state that right there. Say in lymphocytes, in T lymphocytes, we have a relative luciferase activity of 3 with PCD3 gamma 789. Okay, restate it. Say if those were to be placed into kidney cells, your idea on that, whatever you choose, you got to say something similar and say, well, the lymphatic system is a system of vessels throughout the body to clear out unwanted waste. Well, what do the kidneys do? The kidneys are connected to the blood, which is a series of vessels throughout the body, which clears out waste. So to me, I'm thinking they're pretty similar cells. Now, literally, if you write all this stuff out, that's a pretty reasonable, I don't know if it's true, but it is a reasonable justification for my prediction, okay? Prediction means before you speak, right? Before you know. This is before the experiment, before you go Google it. They're asking you to kind of take a shot in the dark, but then have a reasonable understanding or explanation of like where you came to that knowledge. To say, oh, I kind of know some AP bio, right? So that's what I would say. If you can think of something better than in the moment, go for it, okay? And remember that that's usually going to be your hardest question is your last one. So use your vocab, explain it, cite the data, right? Nice job.